Welcome, everyone. Welcome to the weekly HEALEY ALS Platform Trial webinar series. My name is Sabrina Paganoni. I'm the co-PI of the HEALEY ALS Platform Trial, and today I'm joined by Alison Bullat and Catherine Small, who are our patient navigators. I'm going to give people a few seconds to join because I know it always takes time to bring participants in from the waiting room. So thank you again for joining. I see that quite a few people are attending, and as always, we recognize some familiar names. So thank you to those of you who have been joining this webinar on a regular basis, and thank you so much for all the new members of the audience who are joining us for the first time. So for those of you who are new, the goal of this meeting is really to provide updates, but most importantly, to take questions from the audience. So feel free to type your questions in the Q&A box, and then we will take hopefully all of your questions today. So to start, the HEALEY ALS Platform Trial is a phenomenal collaboration among so many people in the ALS community, including academic centers, industry partners, and a lot of fantastic organizations in the ALS space. So thank you to all of you who are contributing, and thanks for also raising awareness about the trial and inviting people to attend this webinar series. Next slide. And again, for those of you who are new, I just wanted to let you know that at this time, we are testing 4 drugs: Zilucoplan, Verdiperstat, CNM-Au8, and Pridopidine. So the innovative aspect of the platform trial is that in the same trial, we can test multiple drugs. So we're testing multiple drugs, as you can see on the screen. This is an adaptive trial. It's meant to continue to grow, to stay open long term. We're already working on adding number 5, so the 5th drug that will come online in 2021, and we're already working with other industry partners for the next few drugs to be added. So the goal here is to always have enrollment spots available for people with ALS, so that as soon as we finish testing these first 4 drugs, we will have spots for other drugs, so that there's no gap and people can continue to participate. Because we used a common and shared infrastructure, we can do something that would not be possible in a regular trial or a regular standalone trial. Now, because we have this shared platform, we can share placebo data, which is illustrated in the picture here, where you see that essentially in any, for any treatment arm, the randomization ratio is in favor of active, meaning that there's more people on active than placebo. So from a participant perspective, the chances of being on placebo are only 25%, and the chances of being on active are 75%. So for 4 people that enter the trial, 3 are on active and only 1 on placebo. But the reason we can do that, again, is because of the shared infrastructure that allows us to share placebo data so that we can still compare each drug against a large enough placebo group to be able to test whether the drug is safe and effective. At the end of the placebo-controlled trial, we do offer an open label extension for all of the drugs that are tested in the trial. So I wanted to start and show you kind of some arrows that exemplify the participants' experience when entering the trial, because when you enroll for something like a platform trial, because the structure of the trial is a little bit different, again, to provide this shared infrastructure that enables sharing of placebo, the experience is, the flow, I guess, is a little bit different. So everything starts at informed consent. That's very common actually for all trials, where the first visit to the site is called the screening visit, where participants are fully informed of the design, the risks, the alternatives, potential benefits of the investigation. So after somebody signs informed consent, they go through a screening process to make sure that they're eligible to enter the platform trial. If they are, they're then assigned to a regimen. Because we have 4 regimens at this time, everyone has essentially a 25% chance of being on A, B, C, or D. And this assignment is random, and it's kind of equal chances for every regimen. And then after that, within that regimen, after somebody has been randomized to a drug, it's then randomized to either active or placebo, again in a 3-to-1 randomization ratio. So this process can be very quick, just a few days. We do have a 6-week window to complete all of this. And really we try to minimize that. We really try to go from informed consent to randomization more rapidly, but sometimes it may take a few weeks, especially because during the pandemic, we had some supply chain shortages at our central vendor. And so they just weren't able to return the labs as fast as we would want them to. So hopefully this will improve, but that's why we have this buffer of 6 weeks. So, so again, within a few days or a few weeks, participants are then randomized and initiate the placebo-controlled trial, which means the period of time where they're tested, but we don't know if they're going to be on active or placebo. And so that period lasts 6 months. At the end of that period, participants who complete the placebo-controlled portion of the trial, they are offered the option to enter an open label extension. That means that everyone who has participated and completed the 6 months of placebo-controlled testing, they can enter, they can receive the active drug for sure for several months at the end of the placebo-controlled trial. That's true whether somebody was randomized to active or placebo. It doesn't matter. In fact, we don't know if people have been randomized to active or placebo, and we cannot know that until the very end of the study. And by very end of the study, I mean when everyone has completed the 6 months of placebo-controlled trial, because it's at that time after the placebo-controlled trial has been fully completed that we can tell if each of these single drug is effective or not. Now, the good news is that because they've been started sort of at the same time, especially the first 3 regimens A, B, and C, we will have sort of a, there's going to be an exciting time, I would say, when we can reveal the answer, really the results of not 1, but 3 drugs at the same time, zellucoplum, verdiperstat, CNMU8. Pridopidine was added just with a short delay, so hopefully they'll be able to catch up, and then we'll get the results slightly afterwards. But again, they're only behind by a few weeks again, and so we'll know the results after that. If you can go to the next slide, that's those are numbers that we track very closely every week. First of all, these are amazing numbers. This means that there are hundreds of people with ALS that have been participating and are participating in this trial. So I just want to start with that acknowledgement that this trial could not be possible without the extraordinary participation of so many in the ALS community. We have over 600 people with ALS that have already signed informed consent, sort of starting the process, as I explained on the previous slide. We have over 400 individuals that were assigned to a regimen or even randomized within a regimen. So that 429 is the number of people who have been assigned to either active or placebo. Again, we don't know if they are on active or placebo, but then we know that they are completing basically the 6 months of placebo-controlled testing. In fact, 128 of them have already completed the placebo-controlled trial, and they are now in the open label extension for any of the available regimens. To break this down by regimen, you can see that A, B, and C have the highest enrollment numbers, again, because they were started at the same time in the summer of 2020. Each regimen, I should have mentioned that, the goal is for every regimen to have a total of 160 participants. So as you can see here, B and C already have 135 or 136 participants each. So that means that, you know, we are really making great progress towards the magic number of 160. So really we're getting close to full enrollment. Now, once participant number 160 has been assigned and randomized within those regimens, then we will still have to wait for the last participant to complete the 6 months of testing. So it's only after everyone has not only been enrolled, but also has completed the 6 months of testing that then the placebo-controlled trial will be fully completed. Of note, regimen A looks like they have fewer participants, but it's really just because they are a couple of weeks behind because there's a two-week vaccination period for regimen A, but really they have the same number of people that have been assigned to that regimen as B and C. And as I mentioned earlier, D is slightly behind because, again, they started later, but then once A, B, and C are fully enrolled, then more people will be sent to D, will be assigned to D, and so they should be able to catch up. So again, we really can't wait to enroll the last few participants, because at that time the 6-month clock will start, and we will know for sure when we're going to really complete the trial. Next slide. So Catherine and Alison, who are here with us today, are our patient navigation team. Please feel free to contact them about any questions. I know these are sort of short webinars, and while they are recorded and the slides are shared, and you can always refer back to them, Catherine and Alison are also available to talk to you about any additional questions, or if there was anything that wasn't clear, please contact them and they can get back to you. Next slide. So we have a great list of upcoming guest speakers. We are scheduling more. The idea is to have site investigators from local sites come and present and tell us more about their sites. We had 4 so far. I think it was a lot of fun to get to know each site. We were able to travel virtually to their site to different states, and so that's the schedule. Next week we're going to have Dr. Jeffrey Rosenfeld from Loma Linda University, is one of our site PIs, is enrolling participants in Loma Linda, California. So please come next week, and especially let your friends from California know, so that we can discuss sort of enrollment opportunities in that state. And we're still working on the biostatistics series, so that's going to come up. But in the meantime, we will be talking with different site PIs to learn more about their sites. And on July 22nd specifically, we will also have Dr. Bedlack join us and talk about his site, Duke University, and also about expanded access in particular. Next slide. For more updates, these are our links. I want to remind everyone that we do have a series of recorded webinars, including webinars regarding the science that supports the drugs tested in this trial. And then next slide. And this is just a reminder for a parallel series of webinars that are available through NIELS. And so NIELS is the largest clinical trial consortium for ALS sites. And they have a great website with a lot of educational webinars that are available for free. So please refer to them as well. And that was our main update. So now I think we can open this up to questions. And maybe Catherine or Alison, can you help me with the take the questions from the audience?
Sure. So we did have a few come in already, and we'll start with the first one here. Someone's asking, how do the shared placebo data, or how can it be significant if the drugs are different? They have different side effects and different drug targets, et cetera.
So that's a great question. So if somebody is on placebo, we don't expect that placebo drug, whether it's an injection or a pill, to have a different effect. And so the placebo data should be comparable across different regimens. Now, on the purely statistical on the purely statistical side of things, there are some adjustments that are made on the models to really make sure that that assumption is confirmed. And maybe that would be a great question for when we have the statisticians. But essentially, the more general answer to your question is that it shouldn't make any difference.
Another question here is, will assessments and communication of the results for regimens A, B, and C have to wait until regimen D is complete, or will the results be communicated as soon as each arm finishes?
Great question. So results will be communicated as soon as each regimen completes. That's a kind of a general statement for the platform trial. However, for the first 3, A, B, and C, because they were literally launched at the same time, we expect the results of all 3 to be available at the same time. Now, there might be a 1 to 2 week sort of difference between them, but it's going to be roughly the same time. So A, B, and C will be announced first, and then D will be announced hopefully shortly thereafter.
And then I think we had some questions that came in ahead of time, so I want to get to those. And one of them, it's kind of a combined question set here. So do we know what % of people have joined open label extension? And have we learned anything new from open label, or are there any side effects that we're finding have to be handled in the open label?
No, that's it. Those are all great questions. So in terms of the open label extension, now we have over 100 people enrolled, which is really exciting. We are learning a lot from the open label extension. We know that the vast majority of people that completed the trial, over 90%, opted to go into the open label extension. And essentially right now it is providing a lot of useful information. However, just as we're doing for the placebo control trial, we won't analyze the results as a whole until the study has been completed. So we will have to wait for that time. Also, there's going to be an analysis at that time for the open label extension as well. So I don't think we have noticed any side effects of particular concern. I mean, obviously, we are monitoring the safety of all participants very closely. So far, we haven't had any sort of concerning signal.
And I kind of wanted to jump off of that. So I've had a couple of phone conversations, and that question actually came up this week a lot, is if there are any side effects that the study PI or study team notices that a patient brings up. What does that conversation look like, or how do you deal with that when that arises?
Yeah, it's interesting you're asking. I had that conversation today with one of our participants, and the truth is that, in the context of the placebo-controlled trial, we truthfully don't know if somebody was on placebo or active, and for that reason, really, we don't know if that particular side effect is related to the drug or not. Now, there are some side effects that are expected for each drug, and so that could be a clue. Having said that, really, one of the reasons the placebo-controlled trials are important is also because at the end of the trial, we need to compare active to placebo and be able to separate the side effects that were truly attributable to the drug from the ones that were just, perhaps frequent, but not related to the drug. Because, obviously there are, common side effects like headaches or nausea or things like that can happen to anyone really in the population, irrespective of being taking a drug or not.
We've had some more questions come in from the audience here, and someone's asking, since one regimen has quite different route of administration from others, could there be a subtle bias there? And if so, how do you control for that?
Oh, great question. So this is really something that has come up a lot in our conversations with the statisticians. I'm going to defer the more technical answer to the statisticians when we have the statistical series. So please come back for when our statisticians will be here. But I can tell you that essentially there's a couple of things that we have done to make sure that there is no bias from that. So number one, the routes of administration, while potentially different, we have a subcutaneous injection, we have a couple of oral medications. They're not expected to be major for these specific routes of administration that are not invasive. Now, if we were to consider surgery, for example, or a more invasive administration of a treatment, such as injection into the spinal cord, for example, then we will have to deal with that. And probably the placebo data could not be shared between people that are on less invasive sort of routes of administration and people who have, who undergo surgery or complicated injections. But, we also chose the drug so that, there would be no expectation of a bias from that. Because again, the drugs that we have, are all delivered in ways that are not considered, too invasive. Now, on the statistical side of things, there are actually the models continue to monitor for that. And there's a lot of statistical work that's going on behind the scene to make sure, to confirm that basically our assumptions are correct, that indeed there is no difference between the different placebo groups. So all of this is handled also on the statistical side to make sure that, everything I told you remains correct and there's no bias. But this is a great question, and we certainly can explain them more when we have the statisticians on.
That was very interesting. Somebody else is asking, how long in general, plus or minus a month, do you think that statistical analysis would take for regimens A, B, and C once it's completed?
Yeah, so we mapped this out very specifically, and essentially it's not just a statistical analysis. There's a number of procedures that need to happen, and everything has already been mapped out. And essentially, I can tell you, I mean, again, give or take, for the first 3 regimens, A, B, and C, it's going to be less than 3 months to have all the results. But again, this is, this is based on a number of different steps that need to happen in sequence. But again, we are already, believe it or not, working very hard on the closeout procedures, even if we haven't really finished the study yet, but because everything needs to be very well planned, and we're already working on that.
I think someone here is kind of asking, looking toward the future of maybe possible future regimens. Do you think that CRISPR and stem cells are possible solutions?
Well, I think that those are certainly things that are very interesting, and I really hope that they will pan out in the future. So I think we continue to review all the science, preclinical science, and new drugs. Actually, there was another question about sort of an antibody-based treatment for congenital myasthenia, which is a different disease, and the question was about using these new methods in ALS. Well, I can tell you that, again, I don't know exactly if that particular treatment will be applicable to ALS. I don't know if CRISPR or stem cells will be specifically applicable to ALS, but I can tell you that we're certainly looking at all of these news, and we have a very active scientific committee that really pays a lot of attention to all of these news, and again, continues to look at the next drugs for inclusion.
And someone's asking here, do you think that the phase 2 rescue ALS trial results that are being conducted in Australia will impact the HEALEY trial results?
That's a great question. Well, as you know, the Rescue ALS trial is expected to finish by the end of the year, and that's a phase 2 study. So it is a little bit different from our study in terms of sort of the size, the design, and the goals. It's very much based on neurophysiological assessments, which are very interesting techniques to really look at the electrophysiology of the motor neurons. So I think it will definitely add a lot of value, and I think that what's really going to be powerful is to see the combination of those results with the results from the HEALEY trial, because we're not taking those measurements. So again, the 2 studies are not redundant, I guess. They are very much complementary because they're very different in terms of what they're measuring. And so I think actually it's going to be very exciting, in the fall when we have the results from Australia, we look at the neurophysiological measures, and then we complete the study here. Then again, it will really paint a complete picture of CNM-Au8.
And I want to go to another question that was submitted ahead of time. So someone was asking to just kind of explain a bit about how enrollment works. When we're partnering with 50 plus sites nationwide, do certain sites have a certain number of spots available for the trial, or how do they work together to get those 160 in each regimen?
Yeah, no, that's a great question. So, so first of all, I just want to make a premise that the number of spots per site, there's no predefined number of spots per site for this trial. But I also want to let people know that there are other trials where it is predefined. So again, what we're describing here is in the context of a platform trial, but there are different models for very good reasons. Actually, there are some trials where they're smaller, they go by cohort, they can only enroll a very small number of people with a specific sequence. So those different studies, not the platform trial, they do have limited numbers of spots per site. In the platform trial, we don't have that. So maybe that's why kind of the question, the question came because perhaps for other trials, there are limitations. For this trial, we don't have limitations. In fact, every site is welcome to enroll as many participants as possible. Now, obviously, at some point, there is a limiting factor, which is the essentially the capacity at the site or the bandwidth. So traditionally we haven't had many ALS trials. Like if you look at the last 20 years, there were a few, but not so many spots available at any given time. So now we have really changed that with the platform trial. So again, many other trials have a very small number of spots, very limited per site. So the site resources historically have been developed to support those sort of small studies or small number of participants per site kind of study. And here we're really changing it. And so I think there's a need for sites to sort of catch up a little bit and increase their resources so that they can really deliver in terms of large numbers of enrollment. So I think we're working very closely with the sites. They're all aware of this. We have regular calls with all the clinical site coordinators, all the site PIs. Many site PIs actually come as speakers, as just mentioned, on this weekly series, so they are all aware about sort of the progress of the study. When the spots are completed for the first few regimens, everyone will be notified so that we can enroll the other regimens, and so everyone will always be on the same page.
And someone was asking if the same placebo group will be used for regimens moving forward, or does the placebo group end when regimen D ends and then a new placebo group will be needed?
Great question. So all the placebo data stays. The plan is to keep it going so that, that will help us in two ways. Number one, as we accumulate a large placebo bank, the hope is that in the future, there will be less need of a placebo. So perhaps we can change the randomization ratio from 3:1, as it is now, for example, to 4:1. That's the hope. So again, we need to first accumulate all the placebo data to be able to do that. And then the placebo data will also serve as really an important resource for the entire ALS community to do more biomarker research, where, as mentioned in previous webinars, we are collecting a lot of biomarkers from all individuals. That's really going to be very helpful, especially in the placebo groups, to really tell us more about the behavior of some of these novel methods, so that hopefully in this adaptive trial, we can adapt, improve the placebo active to placebo ratio, and also employ new biomarkers more effectively.
We have a follow-up question about enrollment methodology. Someone is wondering if enrollment is blocked by certain eligibility criteria, whether it depends on ALS FRSR scores or disease duration.
Yeah, so there are some eligibility criteria, not so much the ALS-FRSR score, but there are eligibility criteria related to time since onset, which is one of the ones you asked about, as well as respiratory function. So those criteria are publicly available. I think we had a link in the slide deck that can be, again, downloaded from the website, or you can also call Alison and Catherine for more information. We also have an eligibility check tool on the website. Perhaps, Catherine, if you could post, or Alison, the eligibility check sort of tool in the chat, so that people can take it and go and check there. So essentially, there are ways to do sort of a first pass in terms of assessment of someone's eligibility. So the reason why we have these criteria is because we have, there's really a need to have good statistical power to see a treatment effect. So really the goal of the trial is to be able to tell as quickly as possible if all of these drugs are effective. And in order to do that, we need to enroll a population with certain criteria so that we limit variability between participants, and that helps us tell if the drug is effective more rapidly. However, if the drug is effective in that population, we expect it to be effective for everyone, not just the people who were originally included. And then I think there's a follow-up question about if the inclusion criteria were blocked in terms of achieving balance across ALS-FRSR score. I'm not fully sure I understand your question, but I guess you were asking whether perhaps there was kind of a cap for certain groups. I'm not sure what you meant by that, because basically, as long as all the people, and feel free to type more in the chat if I'm not understanding, but essentially the reason, as long as we enroll that population, then because we have a randomized placebo-controlled trial, I guess we expect the different characteristics to be equally distributed across, the two groups, active and placebo. So we don't need to do additional sort of balancing. Yeah. Okay, so I think there is one last question that just arrived.
There is. It's a long one, so give me a second to see what this one is here. Okay, so the question is, I'm just going to read it here. Let's say that a trial has outstanding results. It meets all the primary and secondary endpoints. Has the FDA given any indication that they might approve a regimen straight out of the HEALEY trial? Or would they make a company do another phase 3 trial? They're asking because of the results from AMX 0035 and that moving into a phase 3.
Right. No, this is a great question. So I can tell you that we had discussions with the FDA and actually planned the trial design in a way that it could support the registration of a trial. However, one does need to have outstanding results, including results on the primary and perhaps some of the secondaries. Now, there aren't sort of clear-cut guidelines in terms of, you need this p-value or you need that secondary. So that's where sort of the review process comes in. So in other words, we know that the design is such, and the design has a lot of features to that, including the statistical methods. So the design and the statistical methods are such that they could support the registration of a new drug. But obviously that depends on the results themselves. So the actual results on the primary and secondary. Yeah, and I think I hope I answered that question, and happy to continue maybe next week. I know somebody was asking if spinogenics can be considered for the HEALEY platform. Again, we're talking to a number of partners. And actually I don't remember the full list, but essentially, if this company is interested, absolutely they can contact us. Perhaps we are already talking to them again. I don't know. We're talking to several partners, and really this is a great collaboration with so many, and people continue to send ideas and propose new companies and new compounds, which is great. Great. Thank you so much. And we will see you next week. And we will have also Dr. Sukovic and Dr. Rosenfeld from California. So that's great. Thank you so much. Bye-bye.
Thank you.